The next one, metabolism of Xenobiotics, Xenobacilla. There's another word for that, Xenobiotics. This one is Xeno, but another is Xenobiotics, Sino, biotics. That means biotics coming from China. And that's why they call their vaccine Sinovac, Chinese vaccine. No? Okay. Importance. All right, Xenobiotics. Xeno meaning xenos in Greek or foreign. Anything that is foreign in the body that you introduce, anything. No? So this includes medicines, food additives, environmental pollutants. Environmental pollutants, whether we eat it or we inhale it. Or we can also consider sound as a pollutant. No? All right. We encounter many xenobiotics that must be metabolized before being excreted. Okay. Drugs are considered xenobiotics. It is always said before, either this drug is legal or illegal. Then you have chemical carcinogens or compounds in plant foods, and a wide variety of compounds that have found their way into our environment, like PCBs, biphenyls, these biphenyls, and insecticide, and other pesticides. Now, you find this in your plastics. A lot of marine animals now have died because of plastics. Because of plastics that goes into the sea. And degradation there is very, very slow. Sometimes they are eaten as a whole, or sometimes very small particles of plastics are eaten by small fish. And since that cannot be metabolized, they stay in their tissues. And these small fish are eaten by big fish. The big fish now eats also the plastics that these small fish have been eating. And of course, this can be a hindrance to their metabolic processes. So we can just can say that in the long run, if plastics will be around with us and it usually goes into the sea, we may lose all our marine life, in which a large number of people are dependent on marine food. Okay. Aside from that, the usual ones that we take in are these chemical carcinogens that are added into our food, and insecticides that we inhale, pesticides, all of that. And your main organ for metabolism of these xenobiotics is the liver. Now, in the liver, a xenobiotic is being handled by making these compounds water soluble. And if they are water soluble already, they can be excreted in the urine. So there are two phases in the liver, phase 1, phase 2, for making this compound water soluble. So we call them phase 1 reactions of the liver and phase 2 reactions. The phase 1 reaction is hydroxylation of these xenobiotics. So you know hydroxylation, putting an OH. All right. Putting an OH into a compound does not necessarily mean that you have already removed the potency of that compound. Sometimes hydroxylation reactions may make the compound more potent or more toxic. That's why there is the need for a phase 2 reaction. The phase 2 reaction adds compounds to the xenobiotic so that the xenobiotic becomes now or rendered less toxic or non-toxic and is water soluble and excreted through the urine or through your kidneys. No? Okay, here we are. Two phases, phase 1 reaction. Okay, phase 1 reaction. You have a group of enzymes. This one, cytochrome P450. This is quite a versatile enzyme in the sense that it does not only perform hydroxylation reaction or putting an OH, but they can do all the reaction like deamination, removal of amino group, dehalogenation, removal of a halogen. You know what are halogens, di ba? Bromine, fluorine, fluorine. Okay. And desulfuration over of a sulfur, epoxidation, peroxygenation, and reduction reaction. You see? The enzymes that we mostly know is usually perform only one type of reaction. And sometimes some enzymes are very, very specific only for one substrate. But this cytochrome P450 is very versatile. We mentioned already this deamination, the halogenation, the sulfuration, etc., aside from hydroxylation reaction. Okay. Cytochrome P450. The cytochrome P450, there are about, so far, they have seen is 57 of them. 57 cytochrome P450. All right. So that's phase 1 reaction. So let's look at phase 1. The main reaction involved in fish 1 is hydroxylation, brought about by a family of enzymes known as monooxygenases, meaning they add an oxygen. No, mono, only one. Okay? They are also called cytochrome P450. And there are 57 cytochrome P450 genes that they have seen in the human genome. Cytochrome P450, just like the other cytochromes that we mentioned in your respiratory chain, is a heme enzyme. Why 450? Because when it was discovered, it was having an absorption peak of 450. Okay, so they prepared a microsome from the endoplasmic reticulum, chemically reduced, then exposed to carbon monoxide, and had an absorption peak of 450 nanometers. That's why they are called cytochrome P450. At least half of the common drugs that we ingest are metabolized by isoforms of cytochrome P450. What do you mean by isozymes? What do you mean by that? Isozymes are enzymes that catalyze the same reactions, but they are not the same in structure, no, and components. Okay. Isoforms of cytochrome A450, they also act on steroid hormones, carcinogens, and pollutants. In addition, cytochrome P450 in the metabolism of physiological compounds, for example, synthesis of steroid hormones, and the conversion of vitamin D to its active metabolite, calcitriol. Huh? All right, so that's phase 1. Oh, wapa, moment. So this is the overall reaction catalyzed by cytochrome 450. You have a compound, reduced compound. You have an oxygen, and again NADPH. And then the compound becomes hydroxylated. Okay, so if this is your xenobiotic, that xenobiotic now is hydroxylated, and you form molecule of water and the oxidized form of NADPH, NADP. So you can see this reaction in chapter 12, figure 12-6. Cytochrome P450 reductase. Okay. Another one, cytochrome B5, another hemoprotein found in membranes of smooth endoplasmic reticulum, is involved in electron donor in some cases. Okay, let's look at our, okay. So we have isoforms of cytochrome P450 from a superfamily of heme-containing enzymes. Oh, there is systematic nomenclature. So the root name is CYP. This followed by a number of designating the family. Cytochrome P4 are included in the same family if they exhibit 40% of amino acids, is followed by a capital letter indicating the subfamily. Okay. And are the same if they exhibit more than 55% sequence identity, meaning the amino acids are more or less similar, 55% sequence identity. The individual 450s are then assigned numbers in their subfamily. Okay, so here we are, cytochrome pair. Okay, it's the first, and then subfamily A, and the first of the subfamily A. Cytochromes are found in the liver cells and enterocytes. What do you find enterocytes? The word entero meaning intestine. Okay? So you know where they are now.
I'm studying bio and Institute Heart engineers.
Okay.
Not organ engineer is but this is what. Hold on. You can call me Greg.
Okay.
Are you already aware for what I have? What?
Okay. Overlapping specificity of cytochrome P450 explains interactions between drugs, drugs and nutrients, no? Okay. Most cytochrome P450 are inducible. Meaning this can increase if you give a compound that induces its formation. So they are inducible. Okay, an example, phenobarbital can increase the formation of cytochrome P450 within a few days. In most cases, involves an increased transcription to messenger RNA. So this is in the endoplasmic reticulum. No? So there is a threefold increase cytochrome P50 if you give phenobarbital. All right. Now this usually brings about drug interaction. No? Like for example, a patient is taking blood coagulant, coumadin sodium, coumarin or coumadin sodium. Another name is warfarin. And this coumadin sodium or warfarin is used in or are components of a rat poison. That's the common rat poison that you know. Dora, no? Dora. It contains warfarin. It's a blood anticoagulant meaning blood thinner. So blood thinner makes your viscous blood thin out. That's warfarin or commodity sodium. You use that for poisoning rats. Now, usually when you do not use warfarin, you poison a rat with another poison, and you know rats goes into a hole. And if they die in the hole, and it smells, no? And you do not know where it is. With commodity sodium or this warfarin, the rats who take this will go into hemorrhage, internal hemorrhage. And when an animal is in hemorrhage, there is always that feeling of thirst. So the rat comes out of the hole from where they come from, and then they drink water. They drink a lot of water, and they die. So they die outside of the hole where they are staying. You see? That's your warfarin. Okay. So this warfarin is also used in blood thinning of a person, especially if it is hypertensive and the blood is viscous. So you usually give a blood thinner, because a very potent blood thinner is commodity sodium. Now, there is a less potent blood thinner, and that is your common aspirin. The common aspirin, that's a blood thinner. Okay, so what has this got to do with the interaction with cytochrome P450? And it says here, phenobarbital, when given, will induce the formation of cytochrome P450. Okay? Now, if you have a patient who is on warfarin or blood thinner, okay, and then he needs to be given phenobarbital, needs a barbiturate. Naturally, cytochrome P450 will act right away on, because it is induced by phenobarbital, your warfarin will be destroyed right away. So what do? You increase the dosage of warfarin to be effective. Diba? And then, especially if this is in the hospital setting, it says, there is no need for phenobarb for this, so you withdraw the phenobarb. But you did not decrease the dosage of your warfarin or your commodity sodium. So what happens? The patient goes into hemorrhage, bleeding tendencies. So that's one of the interactions. Okay. All right, so you have it there. Induction of cytochrome P450 underlies drug interactions when the effects of one is altered by the administration of other. Anticoagulant warfarin is metabolized by cytochrome 2C9. Cytochrome P50, this is a family of cytochrome 450, which is induced by phenobarb, will increase the metabolism of warfarin, so reducing its efficacy, so that the dose must be increased the metabolism of some widely used solvents and compounds, also like tobacco smoke, many of which are pro-carcinogen. It is induced by ethanol, so increasing the risk of carcinogenicity. That's another. Or naturally occurring compounds found in food, in fruits, like grapefruit has furanocoumarins, that is a blood thinner, which inhibits cytochrome 450 and so affect the metabolism of many drugs. Some drugs are activated by cytochrome 450, so that grapefruit will reduce their activity and are inactivated by cytochrome 450, so that grapefruit increases their activity. Drugs that are affected include statins, omeprazole, antihistamines, and benzodiazepine antidepressants. So be careful with this, these drugs. Polymorphism of cytochrome 450, this of course very, very low metabolism of nicotine to conitin. Cytochrome 2A6 have been identified a wild type and two inactive alleles. Individuals with the non-alleles of impaired metabolism of nicotine are apparently protected against becoming dependent on tobacco. These individuals smoke less, presumably because their blood and brain concentrations of nicotine remain elevated longer than those of the individual. So there is no craving for nicotine. So there is no need to smoke. So they smoke lesser. No? Okay. All right. That's phase one. Phase two reaction, after the xenobiotic is hydroxylated, they are made more soluble in water by adding glucuronic acid, sulfate, or glutathione. And this is conjugation reaction in phase two. So 5 types of phase two reactions. One is, naglaw-coronidation man eh. That is glucuronic acid, glucuronidation. The wrong spelling. So addition of glucuronic acid. Second one, addition of a sulfate. Third one, conjugation with glutathione. And this is the most important thing for your glutathione to work in your body. Glutathione, conjugation of glutathione. And then it is acetylated, and then it is methylated. And so that makes the hydroxylated xenobiotics in fish one more soluble in water and can be excreted through the kidneys and into the urine. Okay? First reaction is glucuronidation. It's the most frequent conjugation reaction. So glucuronic acid. Where would we get glucuronic acid? We get glucuronic acid from the oxidation of glucose in what you call your uronic acid pathway. Uronic acid pathway, which produces glucuronic acid. Okay. This is also used to make your bilirubin soluble in water. Glucuronic acid to your bilirubin. Medtics are familiar with this. They say when only one glucuronic acid added to bilirubin, you call it bilirubin monoglucuronide. And then if two glucuronic acid added to bilirubin, it becomes bilirubin diglucuronide. And they do test for this, di ba? Okay. So UDP glucuronic acid catalyzed by glucuronosyl transferase, an enzyme that transfers glucuronic acid. And they are present in the endoplasmic reticulum and the cytosol. Molecules as 2-acetyl-aminofluorine, a carcinogen, aniline, benzoic acid, meprobamate, a tranquilizer, phenol, and many steroid hormones are excreted as derivatives of glucuronic acid or glucuronides. The glucuronide may be attached to the oxygen, nitrogen, or sulfur groups of the xenobiotics, the substrate. Okay? That's number one, conjugation reaction. Number two, sulfation reaction. Some alcohols, arylamines, and phenols are sulfated. So you know that already. Sulfation reaction. Steroids, glycosaminoglycans, glycolipids, glycoproteins, no, is the active sulfate. The donor of that is SAM, SEM. Ano? Active sulfate, I'm sorry. It's not SAM, it is PAPS. Phosphoadenosyl phosphosulfate. Phosphoadenosyl phosphosulfate, or also known as adenosine 3' phosphate 5' phosphosulfate. But they usually call it PAPS, phosphoadenosyl phosphosulfate. Okay? Conjugation with glutathione, all electrophilic compounds. The tripeptide glutathione, made up of glutamic acid, cysteine, and glycine. Very important in fish to metabolism. Okay, so here is your xenobiotic. This is your glutathione. And so your xenobiotic now has this. That's a sulfur there. R is the electrophilic compound, 4 classes of the cytosolic glutathione transferase. Transfers that glutathione to the compound, xenobiotic. This is an electrophilic compound. Okay. Then you have two others, acetylation and methylation. So an acetyl group is added to the xenobiotics, and now the xenobiotics has the acetyl group. It is acetylated. Some xenobiotics subjected to methylation. Now, instead of acetyl group, a methyl group is added. And the enzyme is methyl transferase. The enzyme here is acetyl transferase. So this is S-adenosyl methionine. This is your SAM, S-adenosyl methionine, as your methyl donor. Methyl donor. The methyl group of methionine is used. Responses to xenobiotics include toxic, immunological, and carcinogenic effects. Okay. So this is binding to your DNA and RNA and the protein. Cell injury known as cytotoxicity. So the xenobiotic binds to DNA, RNA, or a protein, which is really toxic, resulting in cell death or antigenicity, acting on a protein, bind to a protein acting as a haptin, altering its antigenicity. Also, this is a toxic, immunologic, and result autoimmune disease. Then carcinogenesis. Epoxide becomes dihydrodione. Oh. Are you still there?
Yes, Doc.
Yes,
Yes,
Doc.
Doc.
Yes, Doc.
Yes, Doc.
Okay.
Yes, Doc.
Tomorrow, no class.
Yes, Doc.
And maybe Monday, next Monday, Dr. Barito will be your teacher, and she will have plasma proteins and immunoglobulins. And I think it's Monday, no? So, plasma protein and immunoglobulins. Dr. Barito, I do not know, I am not sure yet whether it's Monday or Tuesday. So if it is not Monday, if she will not be lecturing on Monday, I will be lecturing on Monday. Okay? If she lectures you on Monday, I'll be lecturing on Tuesday. And my topic will be muscle and cytoskeleton. Okay?
Hi, Doc.
All right. So let's end the day.
Yay.
Thank you, Doc.
Okay.
Thank you, Doc.
Thank you, Doc.
Thank you, Doc.